Okay, so welcome back to this next video in which we are discussing the protein kinase C pathway. Okay, right, so we are discussing the protein kinase C pathway, and so far what we've got to is the activation of these GQ/11 heterotrimeric G proteins. Okay, so remember, a GQ/11 heterotrimeric G protein is a heterotrimeric G protein in which the alpha subunit is within this family of G alpha Q/11 alpha subunits. Okay, so there are actually 4 alpha subunits that this could be. It could be a G alpha Q, it could be a G alpha 11, it could be G alpha 14 or G alpha 15/16. So although G alpha Q is the most famous, the others all do pretty much the same thing. Okay, so it's one of them is what this alpha Q/11 means. Right, so we now want to go and see what target the alpha Q/11 subunit in the on state is going to have. Okay, so I'll get another piece of paper and we'll begin this discussion. Right. So, the targets all need to be near the cell membrane. They either need to be integral membrane proteins or they need to be attached somehow to the phospholipid bilayer on the internal aspect so that they can come into contact with our alpha subunit that's in the on state. So let's just draw out our alpha subunit in the on state initially. So here it is. Here is our alpha Q/11 subunit. And it's got guanosine triphosphate bound to it here. Okay, and I'll colour it in red just to be consistent. Right, so what is the target for the alpha Q/11 subunit in the on state? Well, basically it's going to be an enzyme known as a Phospholipase C enzyme. Okay, so we're going to now discuss Phospholipase C enzymes. Now, these are not integral membrane proteins, but they are attached to the inner leaflet of the phospholipid bilayer. Okay, so I might draw one of these looking very similar to how I'm drawing the alpha Q/11 subunit, but it's a very different structure. Okay, so this is just how I generally draw an enzyme, basically, with a nice active site here. So this is our Phospholipase C enzyme, or PLC for short. So P for phospho, L for lipase, and then C for C. Right. So there are many different types of Phospholipase C enzymes. Okay, there are actually 13 different types of Phospholipase C. Now, not all of them are activated by alpha Q/11 GTP. So let's discuss the 13 different types of phospholipase C and which types are actually activated by alpha Q/11 GTP subunits. Okay, so there are 13 phospholipase C enzymes. Now, these are categorized into 6 families to help us make sense of them. So the first family of phospholipase C enzymes, and I might do it to this side, is the phospholipase C beta family. Okay, and this contains 4 members. Okay, so it contains phospholipase C beta 1. Okay, shown like so. Then there will be phospholipase C beta 2, phospholipase C beta 3, phospholipase C beta 4. So it will go all the way up to 4. So that's the first 4. The next family is the phospholipase C gamma family. Okay, and there are 2 members of the phospholipase C gamma family. So there is phospholipase C gamma 1, and then there is also phospholipase C gamma 2. Okay, and I'll just bring this more into the center. Right, the next family is the phospholipase C delta family. Okay, and the phospholipase C delta family has 3 members. It has the phospholipase delta 1. Then it doesn't have the phospholipase delta 2. Instead, it has the phospholipase delta 3 and the phospholipase C delta 4. Okay? Then let's go on to the next family. There is then the phospholipase C epsilon family. And in this family, there's only 1 member. And this is phospholipase C epsilon. Okay? Next family, there is the Phospholipase C zeta family, so we're getting more and more tacky Greek letters now, and the only enzyme within the Phospholipase C zeta family is Phospholipase C zeta. Okay, that's the Greek letter that's used for the famous zeta function in mathematics, which has to do with the distribution of the prime numbers. It's one of the Millennium Prize problems that if you solve, well, I think it's one of the Millennium Prize problems, anyway, I think it is. If you solve it, you get $1 million, and it is unsolved at the moment. Okay, then there is the Phospholipase C eta family, so that was the Greek letter zeta. Okay, this is now just eta, okay, and we're going to have another reminder of the Greek alphabet later when we come to protein kinase C itself. Okay, so in the Phospholipase C eta family, there are two members then. There is Phospholipase C eta 1, and then there is also Phospholipase C eta 2. Now, most of these enzymes that we know of, we don't actually know what activates them. Okay, but before I discuss that, let's just check that I actually have told you 13 phospholipase C enzymes. So we've got 4 here, we've got 2 here, that takes us up to 6. We've got 3 here, that takes us up to 9, 10, 11, 12, 13. So those are the 13 different phospholipase C enzymes. Right, now as I was saying, most of these we don't actually know what activates them. So for instance, the Phospholipase C delta family, we don't have a clue what activates them. The Phospholipase C epsilon family we sort of know. Okay, the Phospholipase C zeta and eta families we don't know again. So these ones have question marks by them. Okay. Phospholipase C beta is the famous family. This is the one that when people just say phospholipase C and then don't clarify, they mean an enzyme within this family. Okay, these are these ones over here that are involved in our pathway. These are the ones which will be activated by alpha Q/11 alpha subunits of heterotrimeric G proteins. Okay. Phospholipase C gamma is reasonably well understood compared to like phospholipase C deltas and phospholipase C zetas and etas. However, it's not activated by alpha Q/11 GTP subunits. Instead, it's activated by receptor tyrosine kinase pathways. Okay, so this is totally different stuff here. And then phospholipase C epsilon is quite curious. It again can be activated by receptor tyrosine kinases, okay, but it can also actually be activated by G protein coupled receptors, but not through the alpha Q/11 pathway. Okay, it's got different pathways involving alpha 12/13, and we're not going to discuss that here. Okay, so for our purposes, the phospholipase C we're talking about here will be one of the 4 phospholipase C enzymes that are within this phospholipase C beta family of phospholipase C enzymes. So it will either be phospholipase C beta 1, phospholipase C beta 2, phospholipase C beta 3, or phospholipase C beta 4. Okay, right. So now we've discussed the different types of phospholipase C enzymes. Let's discuss what they all actually do, and this is common for absolutely all 13 of them, although for our purposes we're only going to be interested in these 4 because these are the 4 we've activated by binding our alpha Q/11 GTP subunit onto them. Okay, so basically phospholipase C enzymes are going to break down a phospholipid that is within the phospholipid bilayer. Okay, so let me start by just reminding you of what a phospholipid actually is. So there are certain criteria that you have to obey to be considered a phospholipid. Okay, whoops, what am I doing? Oh no, that's fine, phospholipid. Okay, so you have to have a phosphate group within your structure. Okay, so let me show you what a phosphate group looks like. So a phosphate group consists of a phosphorus atom at the center, okay, double bound to an oxygen atom, then with 2 alcohol groups coming off either side, like so, and then also a single bond to an oxygen, which has acquired an electron via ionic means and therefore has a negative charge. So this is a phosphate group. So within your structure, you have to have one of these. Okay, the other requirement is that you need to have at least one fatty acid within your structure. Okay, so what's a fatty acid? Well, basically, a fatty acid is just the old biochemist's name for what would now be called a long-chain carboxylic acid. And in fact, we've already seen two examples of long-chain carboxylic acids, which would be called fatty acids. Okay, we've seen myristic acid, the 14-carbon fully saturated carboxylic acid, and we've also seen palmitic acid, the 16-carbon fully saturated carboxylic acid. There is, however, an even better sort of prototypical long-chain carboxylic acid than palmitic and this myristic acid. Myristic acid is actually quite rare. Okay, the most prototypical one would be stearic acid. Okay, and once again, this is the old biochemist's name for a molecule that would now be called octadecanoic acid. Okay, so this is an 18-carbon fully saturated carboxylic acid. So if we draw its structure, here is the carboxylic acid group at the head of the molecule, and then we're going to have 16 methylene groups. So again, I don't want to have to draw 16 methylene groups. So instead, what I will do is draw a methylene group, bracket it like so, and subscript the brackets with a 16 like that. Then we'll have a methyl group right on the end, like so. Okay, and that's octadecanoic acid. Okay, right, or stearic acid as it's also called. So that's the sort of prototypical example of a long-chain carboxylic acid, or a fatty acid. So, to be considered a phospholipid, all you need to have is within your structure, you need a phosphate group and a long-chain carboxylic acid. Okay, right. So, I want to introduce you to the biggest group of phospholipids. They're by no means the only phospholipids that are important within the phospholipid bilayer of cells, but they are the main group of phospholipids that you have within the phospholipid bilayer. And the Phospholipase C enzyme is going to work on one of these types of phospholipids, okay? So that's why we're discussing this. So the biggest class of phospholipids that are present within the phospholipid bilayer are molecules that are called phosphoglycerolipids. Okay? Right, so what is a phosphoglycerolipid? Well, as the name suggests, it's going to have glycerol within it. And all of these phosphoglycerolipids are going to be based on the structure of a molecule called phosphatidic acid. Okay, so phosphatidic acid is a phosphoglycerolipid, and basically it's the core one. It's the one from which all the other phosphoglycerolipids are derived. And once again, I'll stress that all phosphoglycerolipids are phospholipids, but they're not the only type of phospholipids. There are other types of phospholipid as well. Okay, such as the sphingomyelins, which we're not going to touch on. Okay, right. So let's have a look at the structure of phosphatidic acid. Now we're not going to draw out the molecular formula, but we'll draw a nice little cartoon. Okay, so we'll start with the structure of phosphatidic acid, and then we'll see how from phosphatidic acid you can get a more general phosphoglycerolipid. Okay, so phosphatidic acid has as its sort of backbone structure a glycerol molecule. Okay, so this vertical line here, which I'm now colouring in green, this is going to represent the glycerol molecule. Okay, so this is glycerol. And glycerol is the old biochemist's name for a molecule that is more correctly called propane 1,2,3-triol. And yes, although this is a mouthful, it's a fantastic name because it tells us exactly what it is, whereas glycerol doesn't give us a clue. Okay, this tells us we're dealing with a 3-carbon molecule, propane, and then we've got alcohol groups coming off all 3 of the carbons. Okay, so overall the molecule has 3 alcohol groups, 1 off each of the carbons. Now, what we are going to do is we're going to take two long-chain carboxylic acids, which are these horizontal lines in orange here, and we're going to esterify these long-chain carboxylic acids onto the first and second alcohol groups of the glycerol molecule. Okay, so these are long-chain carboxylic acids or fatty acids. Okay, so let me just remind you of how esterification reactions occur. Okay, or how they work. I'm not going to give you an electronic flow diagram. Okay, so here's the carboxylic acid group, and we'll put an R group there to mean an arbitrary side group, basically. Okay, and we've then got the alcohol group of our glycerol molecule. What's going to happen is the alcohol group is going to come off the carboxylic acid group, the hydrogen is going to come off the alcohol group. Those two things are going to combine together, okay, to produce you water. And then what's going to happen is the carboxylic acid carbon is going to link to the oxygen of the alcohol group, and this is called an ester link. Okay, so this is how we're going to attach the carboxylic acid groups of these two long-chain carboxylic acids to the alcohol groups that come off the first and the second carbons of the glycerol molecule. Okay, now that isn't yet phosphatidic acid, this is just diacylglycerol here. What we now need to do to create phosphatidic acid is we need to attach another structure onto the alcohol group that comes off the third carbon. And this is going to be a phosphate group. Okay, so we're going to attach a phosphate group on there, and this structure now is called phosphatidic acid. So let me discuss how we're going to attach a phosphate group onto the alcohol group that comes off the third carbon of the glycerol molecule. Okay, so let's say this is the alcohol group coming off the third carbon of the glycerol molecule. And then let's copy out our phosphate group here. So we've got our phosphorus atom, double bound to an oxygen there, with these two alcohol groups coming off it, and then an oxygen with a single bond, and then that's acquired an electron via ionic means and therefore has a negative charge. Okay, so hopefully what you can notice is that this group here, which I'm now circling in pink, that looks mightily like a carboxylic acid group. In fact, if that phosphorus atom was replaced with a carbon atom, that would be a carboxylic acid group if you ignore the fact that it's got 5 bonds coming off it and just look at the thing that I've circled in pink. That would be a carboxylic acid group. So in fact, this can react with alcohol groups in exactly the same way as a carboxylic acid group would. Okay, so what's going to happen is the alcohol group will come off the phosphate group, the hydrogen will come off the alcohol group. Those two things will be combined together to make water. Okay, so these are going to combine together to make water, and then we will bind the oxygen of the alcohol group to the phosphorus atom of the phosphate group, and this is analogous to an ester link, and it's called a phosphoester link. Okay, so we can link phosphate groups onto alcohol groups via these phosphoester links, basically. Okay, right, so that's how we've stuck that phosphate group onto the third carbon of glycerol's alcohol group. Now notice something. This phosphatidic acid molecule is a phospholipid because it's got a long-chain carboxylic acid in its structure and a phosphate group. Okay, so it does meet the criteria for being a phospholipid, and you do have some of this within your cell membranes. Right, okay, so before going on to the more general phosphoglycerolipids, what I want to discuss is why this molecule is considered an acid and what the difference between phosphatidic acid and phosphatidate is. Because when people talk about phosphatidic acid, often they will talk about phosphatidate rather than phosphatidic acid, and I want to just tell you about the trivial difference between the two. Okay, so basically you will notice that this phosphate group that we've got attached onto the alcohol group of the third carbon of the glycerol molecule, it actually has another carboxylic acid-like group still intact within it, even after it has had this phosphoester link formed between it and the alcohol group that comes off the third carbon. Okay, so this group that I'm now circling in turquoise, this is still intact within our phosphatidic acid molecule. Even once we have phosphosterified this phosphate group onto the third carbon's alcohol group. Okay, so what can actually happen is this oxygen here within this carboxylic acid-like group here can donate its proton away into solution, okay, and it will then get a negative charge. That is why this molecule is considered an acid, because it can donate a proton into solution. Any molecule which can donate a proton into a solution is considered an acid molecule. Okay, phosphatidate is the conjugate base of phosphatidic acid. So let me just remind you of what a conjugate base is. So basically, once an acid molecule donates its proton away, use the example of phosphatidic acid, it then is left with a negative charge. For instance, the oxygen here will have a negative charge. Okay, the molecule that you have left is no longer an acid because it cannot donate a proton into solution. Instead, it's actually a base because it can accept a proton from solution onto that negatively charged group. Okay, so a proton can come back and bind onto that negatively charged oxygen. Hence, it is called the conjugate base. And the name for the conjugate base of phosphatidic acid is a phosphatidate. Okay, so when phosphatidic acid gives its proton away, it's no longer called phosphatidic acid. Instead, it's called phosphatidate. Now, under physiological pH, phosphatidic acid molecules do give away their protons and they become phosphatidate molecules, which is why people generally talk about phosphatidate rather than phosphatidic acid, because under physiological pH most of the molecules will exist as phosphatidate rather than phosphatidic acid. Okay, so that's what that difference is there. Okay, right, so we'll call it there for this video, and in the next video what we'll talk about is the structure of a more general phosphoglycerolipid. Okay, so how you can modify phosphatidic acid molecules to create the more general phosphoglycerolipid.